Okay, our job today is to learn about meiosis, but I'm going to start over with mitosis as a reminder and in order to contrast what happens in mitosis with what happens in meiosis. So we've seen this material before. Somatic cells reproduce by mitosis, and those cells are growing or dividing to produce two identical daughter cells. So the actual mitosis itself, where we can see the visible chromosomes, that's actually part of the whole cell cycle. Okay. So overall, the cell will double in size and DNA content, and mitosis occurs producing two daughter cells. Importantly, that the DNA content is the same as the cell that had started the process in the first place. So the idea is to replicate your DNA and produce two daughter cells which are genetically identical to the original cell that you started with. Okay, cell cycle, interphase versus mitosis. The cell cycle has phases G1, S, G2, and M. And we're going to think about that in terms of N chromosomes and C of DNA. So here's our usual depiction of a cell cycle, where we start with G1, which is the growth period, also called gap 1, where the cells are increasing in size, and they are making more proteins, but they're not doing DNA synthesis. There's also a cell cycle stage called G0, where cells that are exiting the cell cycle are ceasing division. Okay, at some point when nutritional conditions are correct, the cell will enter the S phase in which DNA is replicated. Following that, we have G2, which is another phase of growth. And at that point, when the cells are ready, they make the decision to enter mitosis, and then the chromosomes begin to condense, and the actual mitotic division will occur. So it's important to notice that the S phase occurred during interphase, long before any visible chromosomal changes have been seen or can be seen. Reiterating, interphase is when the chromosomes are not visible. Mitosis is when they are visible. And the interphase includes G1, S, and G2. Okay, most of the cells in the adult body are in G0, and the G1 cells are growing, preparing to start replication. And S phase cells are undergoing DNA replication. G2, they've finished replication and are preparing for mitosis. And the M cells are actually when the physical changes that you can see within the chromosomes are visible, and chromosomes are actually segregating from each other. Importantly, the characteristic in terms of DNA content and chromosomes. The diploid cell, remember we're diploid, we have 2C of DNA, which is to say we've got two copies of each chromosome, two N chromosomes, and two N chromatids. Now I think putting some numbers on here makes this a little easier to see. We know that our haploid chromosome number N is 23. So when we start the cell cycle, we have 46 chromosomes in our cells. Each chromosome consists of a single chromatid, so that's 46 total chromatids rather, one for each of the two N chromosomes. So DNA replication is semi-conservative, begins at origins, is bidirectional. Remember semi-conservative replication, each strand serves as a template for the synthesis of a new complementary strand. At the end of replication, each chromosome now has two sister chromatids. So the DNA content doubles during S phase. We go from 2C of DNA to 4C. Importantly, the number of chromosomes doesn't change, just the number of chromatids. So at the beginning of S phase, each chromosome consists of one chromatid. At the end of S phase, each chromosome has two chromatids. So we started off with 2N, or 46 chromatids, and now we have 4N, or 92 chromatids, because each chromosome has two. The DNA content is also doubled. We went from 2C of DNA to 4C. Okay, so I think I just said some of this. The 2C becomes 4C in a diploid cell. Importantly, you still have two N chromosomes at the end of replication, even though each one has two chromatids on it. So don't think of a chromatid as a chromosome. Consider a chromatid as being half of a chromosome that's already finished its replication phase. Okay, so then we start into the actual mitotic divisions. Okay, Okay, remember, DNA replication has already occurred before mitosis. The homologous chromosomes do not pair. Now, you don't need to know the names of the phases of mitosis. I'll say them here. It's not that important to know these. But it's important to know that when the chromosomes then separate from each other, the sister chromatids separate from each other, and that's going to give rise to the two new daughter cells. So here's some pictures. You don't need pictures. Okay, so the actual process of mitosis comes after replication. So when the process starts, each chromosome has two chromatids on it, and you've got 4C of DNA in the cell. So the point of the mitosis is you're going to split that again. You're going to take each chromosome, which now has two chromatids on it, and separate those two sister chromatids from each other. And you're going to therefore produce two new cells, which goes back to the original starting point where you had two N chromatids per cell. Okay, and each chromatid at the end, each chromosome at that point has one chromatid on it. Okay, there is actually a short time during anaphase when there are actually 4 N chromosomes in the cell after the centromeres separate and the daughter chromosomes become, let's say, free-living chromosomes. Right, so here's a picture of anaphase where we're starting off with the sister chromatids both attached to the same chromosome because they were part of the same chromosome, but then they separate from each other. So each chromosome, which previously had two chromatids, now goes back to having one chromatid on it. Your 4C of DNA is going to split. You're going to go back to 2C, and you're back to the starting point where you can start another cell cycle again. Okay, so back to our G1S and G2 terminology. When the mitotic division is finished, we produce two daughter cells, each identical to the original starting cell, only having created a new daughter genetically identical to itself. Meiosis is a different situation, and we have to contrast that to what happens in mitosis, because the job of meiosis is not to produce an identical daughter cell, but to haploidize the genome and ultimately to produce haploid gametes for us. So the point is to start with two N chromosomes, the diploid chromosome number, in our case 46, and then haploidize those, which is to say take each member of a homologous pair and segregate it to a different gamete. So we have to segregate our chromosomes and so that each gamete gets one copy of each chromosome in the set. So the objective is we're going to take two N chromosomes and haploidize that going down to N. So we're going to start off with 46 chromosomes and end up with 23 of them. Okay, so remember, we have two N chromosomes here, which is to say two copies of each chromosome, and we have, therefore, N bivalence, where each pair forms this one structure. So now we're going to have the meiotic divisions. Okay, very importantly, the first meiotic division is the haploidization step, where we have our bivalent, and we're going to take it apart so that the homologous chromosomes segregate to opposite poles in the division. So we had two N chromosomes in the beginning, each pair forming a bivalent. Now we're going to separate those, so that gives us N chromosomes. We still have 2C of DNA, because each chromosome has two chromatids on it, but in terms of the actual number of chromosomes, we have separated each one from its homologue, so now we're down to one each. And that's called the reductional division, when the homologous chromosomes segregate, because that's the one that's going to haploidize the genome for us. So it's important to count chromosomes and chromatids here. After we have the meiosis 1 division, each half of a cell or each cell gets N chromosomes, one of each member of the pair, or 23. Each one still has two chromatids on it, and we still have double the DNA content for that reason. So we're not finished yet. We have to do the equational or second meiotic division, where the sister chromatids separate. So now, when we separate the sister chromatids, and this is very much like mitosis, the two sister chromatids separate, and that's going to lead us to C of DNA in each gamete and N chromosomes. So important to distinguish this. You're going to start off with two N chromosomes, which is to say 46 chromosomes. You're going to separate them in meiosis 1, and now you're going to have 23 chromosomes in each of the products. After the meiosis II division, we still have N chromosomes. We still have 23 of them. But because the sister chromatids separated, we're down to C of DNA. And that's the amount of DNA, the number of chromatids that's going to be present in a mature sperm of a human, for example. Okay, so important to keep track of N's and C's in terms of what's where in this whole meiotic division. Okay, so premeiotic prophase is long and complicated when you think about everything that has to happen then. The chromosomes pair and synapse with their homologs to form bivalents, also called tetrads. Recombination will occur, and it's important that each chromosome find its homolog because that guarantees that it will segregate from its homolog in order to assure that meiosis occurs correctly. All right, so meiosis 1 and 2. Meiosis 1 is a reductional division. Meiosis 1 is a reductional division, and as a result of this, the genome is haplotized. Even though it's still got 2 times the amount of DNA that a haploid genome would have, we have to finish meiosis 2 to get that down. Okay, I think I just said all this. It's important. The haploid nuclei after meiosis 1 still have 2 C of DNA and 2 N chromatids because we haven't had meiosis 2 yet. So meiosis 2 is like a mitotic division in many ways, but it could because sister chromatids separate from each other, as in mitosis, and that leaves each chromosome with only 1 chromatid. So that's called the equational division. The final result, each chromosome, which is to say N of them, has a haploid amount of DNA. Each nucleus has 1 of each of the chromosomes, and therefore has a haploid amount of DNA. It's important to go through this thoroughly to make sure you understand the N's and the C's at each of these steps. This is a good exercise. Draw it. Pretend you have a diploid organism with N chromosomes, and then draw what happens in the diploid cell before replication, after replication, and during the meiotic divisions. There's no way to really understand this until you actually draw it out for yourself, so please do that. Okay, now segregate the chromosomes, replicate this entire process, and make sure you understand what's happening at each step, how many chromosomes, how many chromatids, and how much DNA at each. That's a very important thing to know. A diploid organism has 10 chromosomes in its haploid chromosome set. How many bivalents will be present immediately prior to the first meiotic division in this organism? 10. 1 bivalent for each pair of chromosomes. All the following occur during meiosis and mitosis except separation of sister chromatids, segregation of homologous chromosomes, formation of spindles, chromosome condensation. B. Homologous chromosomes segregate during meiosis, not in mitosis. DNA replication for meiosis occurs once before pairing for meiosis 1, once when the chromosomes are paired, once after the meiosis 1 division, twice, once before meiosis 1 and again before meiosis 2. A. Once before meiosis 1. You don't have to know these, but I would like you to understand what's happening during the process. There are actually 5 stages of premeiotic prophase 1. These are called leptotene, zygotene, pacotene, diplotene, and diakinesis. It's not important to know the names, but to know the order of the processes, I think, is something to think about. Okay, so what we're going to do, remember, we've already replicated our DNA. That happens before any visible changes occur in the chromosomes. The leptotene stage is when the chromosomes first become visible, but they're not yet paired. DNA replication has already occurred. Okay, in the zygotene, the chromosomes are continuing to condense, and they are actually pairing with their homologs, and a process called synapsis is occurring. So there's a proteinaceous substance called synaptonemal complex, which lays down between the paired homologous chromosomes, and recombination is beginning. DNA breaks will be occurring in order to allow recombination between the two paired homologs. Here's a picture of Pachytene cells where you can see them. And then we have Diplotene and Diakinesis. Okay, so Pachytene, the chromosomes are fully paired, and the SC or synaptonemal complex is present between them. Recombination is being completed, and these chromosomes are present in the form of bivalents, each with 4 chromatids, 2 for each of the homologous chromosomes. Okay, so Diplotene, things are starting to pull apart again, and that's where the chromosomes are condensing more. But the important feature of Diplotene is these cross connections between the paired homologs, which you can only start to see because the chromosomes are starting to pull apart a little bit. So these cross-like structures called chiasmata are evidence that the two chromosomes are actually connected to each other because of the recombination crossing over that happened earlier, started earlier, and continues now. So these represent the crossing over events that started earlier in meiosis, but now you can see them cytologically because the chromosomes are condensed. Okay, at diakinesis, the chromosomes are at their most condensed. The chiasmata are still visible, but they move along the end of the chromosome, called terminalization, where they move towards the ends, but they remain holding the chromosomes together. They move towards the ends of the chromosomes, but they do not fall off.